The speakers for this session are Professor Dorothee Viemann and Professor Kaija-Leena Kolho. The chairperson is Professor Alexandra Matias, Professor of OBGYN at the Faculty of Medicine of the University of Porto, and President of the Portuguese Association of Prenatal Diagnosis.
Good morning to you all. I'd like to salute my colleague, Professor Dorothee Viemann. She comes from the Translational Pediatric Department of Pediatrics in the University of Würzburg in Germany. I'm very happy to see someone from Germany, finally. It's very, very an exception in our congresses of obstetrics. It's true. So, she's coming to talk to us about perinatal innate immune programming by S100 alarmins. And I didn't know about this, though I'm a perinatologist, so we are in a very close association with neonatology, and I found it incredible. So really longing to hear you. So you can step
down.
Thank you very much. Okay, so everybody can hear me? Yeah, thank you for this really nice introduction, Professor Mattis, and thank you for the organizing committee. It's a pleasure, and I feel honored to be selected by these young students that this topic might be interesting for you. And I hope I can convince you that it is interesting and imprinting. So the topic is, as you see, perinatal innate immune programming. I put these two pictures here to tell you. Before I have been in Hanover at the Hanover Medical School and led the Experimental Neonatology. I'm still affiliated in Hanover and started in July to this second and now main position for leading the Department of Translational Pediatrics. So this is Germany, this is Würzburg, and this is Hanover, and this is where I'm moving back and forth. What we are talking about today, babies, crying babies. Actually, this I choose because you have to know when I was a student, until my last year, I said like, oh, never ever pediatrics. But at the end, I did it, and it's exciting, I can tell you. Yeah, first I tell you a bit about what motivated me. So this includes also a bit of terminology, and definitions of neonatal sepsis, and especially clinical needs we have. And then we dive into this regulation of neonatal systemic immunity by S100 alarmins, and then the story went on with the role of S100 alarmins in the regulation of neonatal mucosal immunity. Okay, so what motivated me? When I started to do my pediatric education, the WHO announced that actually 44% of overall child mortality happens in the neonatal period. And I found this is an amazing number. And for those that are not so familiar with these terminologies, what is a neonatal period? It's the first 4 weeks of life. So the first 28 days are very critical to dive into this extra uterine world. And of course, naturally, it's very critical for preterm infants. So what is preterm birth? This is when a child is born below the 37th gestational week. You have to know that 7 to 8%, almost 10%, say it roughly, of newborn infants are preterm infants. And 1% of all infants are born with a birth weight below 1,500 grams, which is a critical number. And especially this group is the main reason of death in this neonatal period is sepsis. And neonatal sepsis is different than adult sepsis, very rapid courses, totally different clinical picture in my opinion. And we differentiate between early onset neonatal sepsis, EONS. This starts before the first 72 hours, mostly vertically transmitted. So bugs are involved like group B streptococci and E. coli. They account for 70% of all EONS cases. And more frequent and important for us as neonatologists is this late onset neonatal sepsis. Usually a nosocomial sepsis starts beyond 27 hours after birth and is usually a gram-positive sepsis induced by streptococci. So either coagulase negative streptococci or Staph aureus. And the numbers, the incidences are higher in LONS, so this what matters for us as neonatologists, over 10%, almost 15%. This is still, these are high numbers for doctors. And the mortality is high, still over 10%. So when I was young, I really, like a naive one, I stepped into the lab and said, so I want to clarify what's this was about neonatal sepsis. And what's another very important information for you is only in few cases we have blood culture positive cases. And this is where my first trouble was with. I was thinking, is this all really sepsis? Or is this just something else? Is it just a challenge by microbial colonization or whatever, but not really sepsis? So this is important to have in mind. For comparison, in older children and adults, blood cultures are positive about 40 to 50%. So, and the concept of adult sepsis, this turned out when I was still a student is what makes, what is the pathogenesis of fatal adult sepsis? So fatal cases are characterized by a stage of immunoparalysis. So you have the septic wave, hyperinflammation, cytokine wave, and so on. But when it comes to no reaction anymore, immunoparalysis, this indicates the doctor, okay, now it's really critical. And this is becoming to be fatal. So, and then they adapted a bit this whole concept to the neonates, like usually. They think they are little adults. And the opinion was the immune system of the newborn infant is immature. Okay, there we would agree, compared to adults. And then it comes explaining the high susceptibility of newborns to infections. And this was meant in a sense like we have a primary immunodeficiency. We have impaired inflammatory responses from the beginning. And so we have like a state of primary immunoparalysis paralysis from the beginning on. So, and this explains everything. And this opinion was derived from ex vivo experimental settings. So you take neonatal immune cells, either monocytes, PBMCs, or whatever, you stimulate them ex vivo, and you get lower responses in these typical markers, cytokines, and so on, either in gene expression level or on cytokine protein level, whatever. So this is, we have reduced cytokine responses, we have hyperinflammation, and this is why people thought this is a reason why they are so susceptible. But when I started with pediatrics, really my first position was on the neonatal ward. I saw different things. I thought that neonates, the neonatal sepsis, is a hyperinflammatory state. Fulminant, very rapid causes. Within 1 hour, the child is dead. And again, to remember you, 90% of blood cultures are positive. So this in vitro and this in vivo situation didn't match at all. And so this is finally what motivated me. The story must be different. So let's start, let's see. So again, if you put this in concept, this is the old concept. This is what you see ex vivo, and this is what I saw actually in vivo. So the questions we posed is, this former definition of immature, is this really, or is the immaturity really this immunodeficient part? Or is it something else, what is really immature? And nobody checked really whether these in vitro findings, so when you have a child who have very low responses, is indeed linked to infectious susceptibility. Yeah? So we questioned this. And another important information, a lot of studies showed this, and this is one of the first ones in mice by James Wynn. He showed in mice that the neonatal immune response primarily depends on the innate immune system. So adaptive immune system, this we all know, is immature, needs to be trained, and so on. So they strongly depend on the innate immune system. They just took RAC mice, so adaptive immunity deficient mice, and here you see in adults, RAC mice perform in a sepsis model really worse. When you do the same in neonatal mice, they perform worse in the same manner, and you can't rescue them by adopting, for example, T-cell directed therapies. So for me, it was clear if I want to explore neonatal sepsis, I have to focus on innate immunity. And we started with a very simple experimental setting. We did a systems biology approach. We just isolated monocytes from adults and from cord blood. We stimulated them with LPS from gram-negative bacteria and did a transcriptomic analysis. So we established from these data co-regulated networks, and so you see here the upregulated and the downregulated ones, and we plotted what was downregulated in the adult monocytes and what was upregulated in the neonatal and downregulated. And if you look carefully, you see there are other genes upregulated in the adults than in the neonates, and others downregulated in the adults compared to the neonates. And the question was, why did they use different signaling pathways? So we took all they upregulated in a batch and performed in silico these signaling pathway analysis, and found that in adults, this upregulation is mainly mediated by the MyD88-dependent pathway, the MyD88-dependent TLR4 pathway, because LPS is a TLR4 ligand. And in neonates, upregulation was mainly mediated by TRIF-dependent pathway. Just to remember you, TLR4, this is a receptor for LPS, and it divides into the MyD88-dependent pathway, adapter molecule. This is a TRIF-dependent. This is a very quick one, using finally NF-kappa B to induce a very pro-inflammatory gene program. And this is a TRIF-dependent pathway that via ERF3 induces a regulatory gene program, which at the final end links to the adaptive immune system also. Yeah, so different usage. So this is more used in neonates, and this is maybe more used in adults. So let's see whether this is true. So we looked for, here this is a fold change induction of these MyD88-dependent genes, and they are more induced in adults, and this was long known for long, as I showed you in the previous slides. New was that we could confirm that these TRIF-dependent regulatory genes are strongly induced. So they are not generally not able to respond. Here they actually respond much stronger. And this was a mystery, and to elucidate what's the reason for this differential usage of adapter signaling, we performed this PCA. And you see in this PCA, so these are the adults monocytes after LPS stimulation. These are the neonates, and these are the basis states. And as you see, and then it was somehow crystal clear, it's actually already the basal state that makes the difference of the APS response. So this is like a parallel shift. And so the reason for this different response must already be defined at the basal state. So the baseline programming defines the response. So let's have a closer look to the basal programming. And now plotted onto this co-regulation network of gene programs is the difference between neonates versus adults. That means blue genes here are lower expressed in neonates compared to adults. These are higher expressed in neonates compared to adults. Same plot, but looking at the LPS level, comparing neonates with adults, you see they lose this differential expression pattern a bit. And putting this into another graph, it tells you the following. So here we have genes that are lower expressed in neonates, higher expressed in adults, and after LPS stimulation, they come to the same level, right? Okay. That means that they must have performed a higher shift induced by LPS, these neonatal ones, compared to the adults. So the question was, are these drift-dependent genes? Because there we know these are stronger induced in neonates. Then we have to prove that they are lower expressed at the baseline level. The other way around, these are the genes higher expressed in neonates, end up at the same level, meaning they perform lower shifts higher in adults. So these might be the MyD88-dependent genes. Then we have to prove that they are in a way pre-activated in neonates. And so we had a closer look to these drift-dependent regulatory genes at baseline, and we could confirm, yes, they are almost not expressed at all in neonates. They have no expression tonus yet at birth and perform higher food changes. And conversely, the MyD88-dependent pro-inflammatory program is pre-activated in neonates, resulting in lower food changes upon LPS stimulation. Yeah? So this baseline programming might be connected to epigenetic regulation. And you heard yesterday in this nice molecular biology session about epigenetic regulation. So heterochromatin, when it's packed, it can't be transcribed, and when it's opened by acetylation of histones, then the polymerase is able to transcribe. So acetylation means active transcription. For example, methylation of K4 means, okay, I'm ready to be transcribed. But also methylation of K9 can be inhibitory. And there we had a closer look to this situation of histone modifications in adults and in neonates. And here we took the promoter regions of these pro-inflammatory genes. And what you see is they are not acetylated in the adults, almost not, but upon LPS stimulation, they become acetylated and are consequently transcribed. In the neonates, we found this pre-acetylation, which matches to these pre-activated states, and nothing happens significantly anymore after LPS stimulation. And the picture in drift-dependent genes looks completely different. Here we have this acetylated, means constantly transcribed situation in adults for this program, but no acetylation yet in neonatal cells. Only upon first LPS stimulation, they get acetylated and the chromatin opens. Okay? So we have this different baseline programming, and these are the extremes, the neonates and the adults, and we were interested how is this reprogramming going on, and we looked at these different gene programs. And you see, as we found already in the extremes, that this reprogramming, this pre-activation of the pro-inflammatory genes like I6 and I1 it comes down pretty fast during the first 2 weeks, whereas this increasing expression, baseline expression tonus in regulatory gene programs happens over the first year of life. And some is then completed, and some need further maturation. And this is for me actually what has to mature in neonates. It has to mature that you are able to activate cell autonomous regulatory gene programs to perform regulated immune responses, challenges with the environment. But who mediates this pre-activation? And then I took out of the drawer data I had that I couldn't interpret, which are these S100 alarmins. Now they come into the game. These S100 alarmins, A8, A9, are myeloid-derived proteins. Actually, 80% of the protein production of myeloid cells is done for S100 alarmins. And when they are released, they perform these, they form these heterodimer complexes. And we found high levels in healthy term infants. And this is important because this S100, A8, A9, which is also called calprotectin in the clinics, in adults, it's a perfect biomarker for inflammation. But these are healthy ones, not inflamed, not ill, not at all. So in former days, I couldn't interpret it. But what we found in this first study on this is, since it is TLR4 ligand, if you pre-activate or pre-treat or pre-prime cells with this endogenous TLR4 ligand, following exogenous stimulus like LPS to the TLR4 ligand isn't able to mount such an inflammatory response anymore. So it's a kind of tolerated system compared to unprimed S100 naive cells. So here full response, here a tolerated LPS response. So I thought this might be the concept in the neonate then, and this might be the job for these S100s. And so we looked for in these neonatal monocytes and cultivated them for a longer time ex vivo and without, so being out of their plasma, being in the culture setting, without any S100 anymore around. They lose this pre-activated states in these MyD88-dependent genes. If it's cultured in the presence of S100, they keep their state. And S100 has no influence on the TRIF-dependent genes. This is important. They don't induce them. And this is because they actually use another binding site in the groove of the TLR4 receptor than LPS, for example. Okay? So can we use this? Can we exploit this whole finding now for the clinics? And first we tested it in a mouse model of endotoxemia, meaning LPS. This is not it's more a CS model than a sepsis model, as you know. And we pre-treated neonates with S100, one single shot after birth. Then 2 days later, they got LPS. And then we looked at the performance. And as you see, so if you are S100 deficient in knockout mice, you are very susceptible to fatal causes of endotoxemia compared to white types. But you can rescue them with different treatments, S100 homodimer, or with the heterocomplex, and S100 A8 dimer is even more potent than the complex. In the meanwhile, we have explanations for this. I won't dive into the high biology of S100s today. And of course, it needed to be also performed in a real sepsis model. So this is an S. aureus-induced neonatal sepsis model. And again, the S100 knockout mouse is very susceptible and has a survival disadvantage. And we could rescue them with different treatments of S100 alarmin preparations. But since I'm a neonatologist and not only an animal researcher, we also looked in our cohorts. In term infants, the levels are higher, significantly higher than in preterm infants. For me, then at that stage, no wonder, because they are more susceptible to sepsis. And then we looked in this preterm group and stratified them into those who got sepsis, late onset sepsis, and those without sepsis. And there we found that those that got sepsis had in their cord blood significantly lower levels than those that got later no sepsis. And this was amazing. This turned out that this is an odds ratio. If you have a level below 330, which is a normal adult value, then you have a risk, a 25 higher risk to get sepsis than if you have a normal adult value. These are numbers that are for medical doctors amazing. Now I try to summarize a bit the concept. It's important for the mucosal immunity. So this is the adult situation. Low levels down in adults for this MyD88 inflammatory program, but you have a certain expression tonus for regulatory programs, so they can control LPS responses. It's a bit also induced this regulatory program after LPS, but this induction of inflammation can be controlled here. So in term neonates, in neonatal physiology, it looks like this. So we have an immature state expression of regulatory genes. They won't be able really to control these inflammatory responses, but good luck, they have high alarmin levels that do this priming and S100 mediated tolerization, so no strong responses here. But for example, like in preterms, where we have low levels of S100 alarmins and even more immature expression tonus for regulatory programs, there's nothing that regulates such inflammatory responses. So we have hyper-inflammatory responses at the end. So the result is, at the end, this restriction of the inflammatory response of neonates is not a sign of immaturity, and this was what was new than what we claimed. At the end, it's a meaningful programming to manage this clash with the new environment. And it's mediated by S100 alarmins, as we found, and in the meanwhile, other people found other things that actually follow the same concept. Yeah? And what clinical implications does this have? I told you, so we have this pre-activation here of MyD88. It comes down, is regulated by S100 alarmins, but this regulation is lost pretty fast. And then we have this rather slower maturation of TRIF-dependent regulatory programs, probably through contact with the environment. At the end, you can read out in the histone modifications, and the net effect of this balance of regulation and inflammation makes up the final response to inflammatory challenges like LPS. And as you see, here is a critical phase. It comes down here pretty fast. This is not yet totally induced, and this is actually what you see as neonatologists. So the peak of sepsis, late onset sepsis, is at 7 to 14 days. Yeah? And this nicely explains why this is a very critical stage, especially period, especially for preterm infants. Yeah. And another clinical implication is we don't only have the outlook for therapeutic usage, you can also use it for diagnostics. So we confirmed this is in another cohort, a huger cohort, that we have low levels in lost patients of S100 in the first 2 days of life, and those that get no loss have higher levels. And we did models, multiple effect models, mixed effect models for all risk factors, for the impact of risk factors on the incidence of sepsis, and turned out that actually the S100 has the highest impact, and even higher than intermittent ventilation, to be a May one, to be small for gestational age, mode of delivery, central venous lines, birth weight, and so on. So this was even amazing for me. And here the odds ratio was, if you have levels below 300, you have a 8-fold higher risk to get a sepsis. But what might be even more useful for a neonatologist is the other way around. If you have levels over 1,000, you have only an odds ratio of 0.2. And this is important because our neonatologists, if you are afraid that this might be sepsis, you use empirically antibiotics. And this concept we would like to leave. But due to this hyper-rapid course, you have no other chance than to protect with antibiotics. And when you realize, okay, it's no sepsis, you stop usually antibiotics. But we would like to get rid of antibiotics at all, you know? Okay, so now we come to mucosal immunity, because when we realized that this S100 is like a handbrake that brings you into life, maybe it's even more important at barrier sites. So, but at barrier sites, so mucosal immunity, there it was like the microbiota, the opinion is the microbiota are the most important trainers. There are good bugs and bad bugs. And important for the establishment of the microbiome is the MOD, mode of delivery, and mode of feeding. So this is a concept, was a concept. But how is it the other way around, we ask? How is it, how does a host and its programming of immunity at the beginning of life, what's the impact on the establishment of the microbiota? So this reactivity might be determined by S100 alarmins. And it was intriguing because it's actually the same concept as the microbiota, using innate immune receptors. It's a TLR4 ligand. So we asked, how should the neonate immune system be programmed to ensure really favorable, healthy colonization? And what's about the role of S100 alarmines? And we found that breast milk has extremely high levels of S100 alarmins coming down during the first months of life. And again, in breast milk, it's very low in preterm infants, and also after cesarean section. And the fecal levels of calprotectin, again, high at the beginning of life, always the same pattern, lower in preterm infants. There it even increases in the first days of life. And again, a dependence on the mode of delivery. So this might explain a lot what we knew, how important mode of feeding and mode of delivery is. Yeah? But the high levels in the intestine are not only coming from the supply of breast milk, so there's also an endogenous production by lamina propria macrophages. So the neonate itself produces S100. And you see here these rare samples of healthy neonatal intestine tissue, small intestine, large intestine, high expression of S100 A8 positive lamina propria macrophages, yeah? A high proportion. Then we went into the mouse models and looked for intestinal immunity here in knockout mice and wild-type mice, and looked for the expression of CX3R1, which is an important marker for the regulatory phenotype of lamina propria macrophages, and it's lower expressed in the S100 knockout mouse. We found it also by immunofluorescence. What's about the pro-inflammatory responsiveness? And there we found a bit the same like in systemic immunity. So this is a baseline programming. This is the response to LPS. And it's overshooting if you lack S100, like here in this mouse. Later on, it's more the alarmin function, the added alarmin function. So when alarmins support inflammatory responses, then it's coming down. But for the regulatory phenotype, the pattern is again consistent. Lower induction and expression tonals in S100 deficient situations of I10 and TGF-beta. TGF-beta, I10, and CX3R1 are very important signals in the intestine after birth to induce the expansion of regulatory T cells. And this we looked up and we found, okay, yeah, so we have a deficient expansion in the lamina propria of the intestine in S100 deficient situations, but this is not a problem in cecal lymph nodes or in mesenterial lymph nodes because they invade from these lymph nodes into the intestine. So it's a local expansion problem and probably due to the lack of the induction of a regulatory phenotype in intestinal macrophages. So this we also confirmed by immunofluorescence. We did ex vivo co-cultures assays of naive T cells with wild type or knockout LPMPs. So and with in co-culture with S100 deficient LPMPs, you induce almost no Tregs, but with wild type this works. Can we use this priming of the intestine with S100? And then we established a model where we fed a single, so it was a single dose of S100 AA to neonates. And then we looked on the phenotype of LPMPs next to all the other markers, so we could revert it in a way here, the CX3R1 expression, and also the induction of Tregs was rescued. And now, does this change of immunity by S100, this priming of intestinal immunity, has an impact on the establishment of the microbiota? And yes, it had. We cohorsed the dams, the wild type and the S100, and followed the offspring and looked, for example, culture-based here for E. coli growth. And you see there's an early E. coli growth already in the knockout mouse that doesn't start in the wild type offspring before the jumper age, we say, so early adulthood. Yeah? And this was a sign of dysbiosis in S100 knockouts. Then we did this cross-fostering and confirmed first in this setup that in the wild type situation, offspring wild type, the fostering dam wild type, no early growth, E. coli growth, but if but a strong expansion in the KO situation, and when the wild type offspring gets KO breast milk, yeah, and then E. coli shows up earlier in life, but you can completely rescue the KO offspring by feeding wild type breast milk. So this was very hopeful. And then we did the same model here, single feeding of S100A8, and this depressed the early E. coli growth in the intestine. And this is not an antimicrobial effect of S100s, because we formally show E. coli growth in breast milk does not change if we deplete breast milk from S100. And S100 homodimers have no antimicrobial effect, only the heterocomplex. And we did the model with S100A8. Yeah? And besides that, the extracellular half-life of S100 is only about 24 hours, so it's a longer effect. Then we looked in human cohorts. So we established this IROC birth cohort in term healthy infants and found, like all others, so we have an increase in the diversity, and this is like everybody else published, an increase, an expansion in Bacteroidea and Clostridia, and so this was all fine. But then we looked in this cohort for the impact of these developments in the microbiota establishment. What is the role of S100? And this is controlled again, the MOD. So with age, we have this expansion, for example, for Clostridia, and this decrease in gamma proteobacteria. And what we found is that the role of S100 has impact on the decrease, contributes to the decrease of gamma proteobacteria. E. coli is part of gamma proteobacteria, and it induces an expansion of actinobacteria, for example, bifidobacteria. So yeah, this was really then convincing that the priming of immunity finally impacts on the development of the microbiota. And the clinical implication again is, so this, we had a look at necrotizing enterocolitis, one of the most feared complications in preterm infants. And you see here in the small and large intestine, high expression of S100 in lamina propria macrophages. And here we looked at the healthy resection boundaries in neck samples, and we have a low expression. So S100 probably there is protective. And in sepsis, same situation like for the systemic levels, also in fecal levels. If they are low, the risk of sepsis is very high. And can we, and this is important for the neonatologist, would we also be able to rescue preterms from sepsis by nutritional supplementation of S100? And yes, this was possible. So this one-time feeding with S100 rescued at least mice from fatal causes, and this was linked to lower cytokine responses, less bacterial burden. So summarized, it's the following. We need high levels of calprotectin in the neonatal intestine to induce a milieu that restricts inflammation and induces a regulatory phenotype in the lamina propria macrophages to promote expansion of Tregs. And this whole milieu at the end allows commensals to survive, to seed, to colonize the intestine, which at the end also restricts the expansion of pathobionts. If we have a deficient situation, you have like an inflammatory milieu, commensals won't make it, pathobionts expand. So at the end, it was like, it's like a concept, all over concept. It's like an ubiquitous, universal regulatory concept. So, and then I might end now with an overall summary, and this might be the main message. The immune immaturity of newborn infants that we previously considered as reason for susceptibility to sepsis and so on is actually, this is a meaningful program state. And this at the end allows this smooth transition into the extrauterine world. Yeah? And maturation, this training and tuning that is required for antimicrobial and regulatory molecular programs, or this is required for regulatory programs mainly, but not really for the inflammatory ones. This, the preterms can do. This is what we see in the clinics. Yeah? So apparently, in this perinatal situation, there are transitional alternative regulatory mechanisms, such as S100 alarmins, that substitute for the lack of cell autonomous regulation. Yeah? And these, at the end, ensure uneventful immune adaptation to the environment. No, I have to thank a lot of people in my groups, in the obstetrician department, the pathology, immunology. So many people were involved in this whole study, including external collaborators. And I invite you to come to Würzburg. I have many open positions because I just started. And thank you for your attention, and I'm happy to take your questions.
Well, very exciting news, very thrilling presentation, and I'm following. So, very interesting. But I think we take questions afterwards, because I think the next presentation has something to do. No. After the next presentation from Professor Kaija-Leena from Helsinki, because I think both are connected, and at the end we could discuss. What do you think?
Wonderful.
It's okay? I was asked by the organization to postpone the discussion today.
Yeah, that's wonderful.
And now, are you connecting to the next presenter? Good. Okay. So, our next speaker is Professor Kaija-Leena Kolho from Helsinki, and she is a pediatric gastroenterologist. So we heard a lot about microbiota and how it is regulated and how important is epigenetics. And so I think we should now listen to what she has to tell us about this healthy early life microbiota in an infant, and afterwards we will do the discussion. Okay? This is a recorded presentation, but I think for the discussion she will be available.
Everyone. My name is Kaija-Leena Kolho, and I'm currently acting as a professor of pediatrics at the University of Helsinki. I'm also a pediatric gastroenterologist, and that's why I have a special interest to gut microbiota. I'm very delighted to be here today and to share and discuss you with some of the recent findings related to gut microbiota and its importance to child health. We all know that our living conditions are not static but under continuous change. This is also true for the disease burden in children. Some diseases become more rare along the years and new diseases arise. As we have experienced an increase in the number of autoimmune diseases in children, there has been a lot of discussion whether the level of hygiene in modern societies is too high and whether children should spend more time outdoors to get contact with environmental microbes, these affecting the priming of the immune system. But environmental microbes do not colonize the human gut. And to make things more complex, autoimmune diseases are diagnosed nowadays more and more also in pets like cats and dogs. So it's agreed upon that environment in which children are living primes their immune system, but we do not know how and which factors are the key drivers. Here you see an example of changing disease pattern. In early 17th, atopic diseases were very rare, but colonization with Helicobacter pylori was frequent. However, after 20 years, the situation among young adults has changed completely. Of those born in early 19th, almost 1/3 presented IgE antibodies at the age of 20 years, when the respective figure was less than 5% 20 years earlier. Observations like this raised the idea that a change in gut microbiota might play a key role in chronic diseases. But it was not until the laboratory methods for microbiota analysis developed that microbiota research leaped forward, and more specifically when one learned to analyze bacteria without culture, and that is with 16S sequencing technology, that the research on microbiota started to progress. Here you see an example of studies reporting that microbiota is altered in a chronic disease. This alteration is named as dysbiosis, but there is no uniform definition for dysbiosis. In general, dysbiosis includes less diversity of bacteria and a relative decrease in most often bifidobacteria and increase in proteobacteria as major findings, and also different clustering of the microbiota profile compared to healthy controls. The methods used vary in different publications, the handling of the samples vary, and the level of reporting the bacterial taxonomy varies, making the comparison of different studies very difficult. However, it's important to keep in mind that most studies are reporting just an association between the microbiota profile and disease, and there are hardly any data on functional interactions. So, regarding the pathogenesis of chronic autoimmune diseases, the current theory is that there is an environmental trigger that changes the balance in gut microbiota, resulting in immune stimulation, and in some individuals, inflammation and presentation of a chronic disease. The other theory is that the function of the immune system is altered for some reasons, and this will result in loss of tolerance to commensals in the gut. That means the normal bacteria colonizing the gut, not pathogens, and this in turn results in a disease process. This figure here illustrates the development of chronic inflammatory bowel disease, Crohn's disease, but similar findings have been reported in several other autoimmune diseases, so that other diseases present also inflammation in the gut. So a key question is, what defines normal gut microbiota and how does it develop in an infant? We do not have a precise definition for normal gut microbiota, but we do recognize several factors that shape the microbiota. As you see in this figure, birth mode, antibiotics, genetic factors, siblings, pets, and diet are all important, but their relative contribution is unclear. Here you see the general developmental pattern of microbiota in early life. After birth, there are huge differences between babies depending on whether the birth was preterm or not, or whether there have been complications or, for example, antibiotic use. But along the first years, these differences disappear, and then the so-called beta diversity decreases, as marked here with a red line. Regarding a single individual, the diversity of microbes in the gut start to increase after birth, and this increase continues throughout the childhood. Previously, it was thought that the shaping of microbiome is complete after a few years, but nowadays most believe that some degree of shaping is occurring throughout life. On the right-hand side, you see some of the major microbial families or orders. Most evident is the high abundance of bifidobacteria after birth and the increase of other bacteria later on. However, it's important to realize that most studies report relative abundances of bacteria, and if a relative proportion of some microbe is going up, something else must go down. This phenomenon hampers the interpretation of the findings in many studies. One needs to decide which one is more important. The observation that some bacteria are going down in abundance, or the observation that some other bacteria families are increasing in abundance. So we will definitely need more quantitative data in the future, but so far there are only few published studies reporting quantitative data of the microbes in the gut. What might be surprising is the similarity of the developmental patterns of microbiota in infants across the world. Here is a summary of published worldwide data, including studies from Europe, North America, Asia, and Africa. And you see the general patterns of different bacteria are quite comparable during the first months and first years of life. However, as said earlier, environment has anyhow a profound effect on the shaping and functional properties of the microbiota. This slide shows an interesting study in which the researchers compared the functional properties of infant gut microbes between Finland, Estonia and Russia. These countries share similar nature, but the living standards have been quite different until recent years, and there is a big difference in the prevalence of atopy and type 1 diabetes in children between these countries. Finland is the country with one of the highest incidences in the world related to type 1 diabetes. Interestingly, in Finnish children, the major bacteria in infant stools were Bacteroides. And in mice studies, these bacteria were capable of inducing diabetes. And vice versa, the major bacteria from children and infants living in Russia possessed bacterial families with lipopolysaccharide LPS production that was considered to protect from diabetes in the same mouse model. So one may conclude that the priming of early microbiota is very important for later disease development, but we should also learn a lot to understand the possible protective effects of the microbiota composition and to be able to use that in clinical practice, but we are not there yet. So one could ask the next question. When does the shaping of the microbiota occur in an infant? It may start in utero, as some researchers have suggested, but it's agreed that birth mode as such has a clear and very important effect. This slide illustrates the impact that caesarean section has on the infant microbiota. There is less diversity in the early days of life in gut microbiota when compared to vaginally born babies. The feeding mode, whether it's breastfeeding or formula feeding, seems to have less effect than the birth mode as such. So the next question is, does this matter? Or what does it mean if there is less diversity in the early days of life in the gut microbiota? There are several reports showing that caesarean section is a risk factor for many chronic diseases. A recent review summarized that the risk is obvious for asthma, respiratory infections, and obesity, but for other diseases listed here, the risk is questionable and not always agreed on. However, you need to realize that this is a statistical difference in large cohorts of infants and that the absolute risk is anyhow low, and most of the children born from a caesarean section are completely healthy. However, the proportion of caesarean sections is increasing worldwide, and therefore it's also important to study various aspects related to this birth mode and infant health and the possible long-term consequences that this birth mode may have. As said before, it's agreed that microbiota is different in vaginally born children compared to those born from a C-section, meaning caesarean section. Therefore, we conducted a pilot study and examined whether it was possible to modify the microbiota in infants after delivery with C-section. And here you see the outline of the study protocol. The study was published this year in a very prestigious journal called Cell. So we chose to perform maternal fecal transplantation instead of vaginal slabs and gave a small amount of maternal stool mixed in the first feeding, that might be breast milk or formula, that the baby received after caesarean section. We of course screened the mother for possible pathogens in her stools before this elective procedure. All the mothers had elective caesarean section. So you may think that it's odd, but the babies drank the milk with no problem, and the transplantation process as such was well tolerated and did not result in vomiting or diarrhea in the babies. And here you see the major results of this pilot study. Those with vaginal birth are marked here with blue line, those with cesarean section with red, and those with fecal transplant and cesarean section with black line. And as you see, the blue and black lines are quite similar, and the red line indicating cesarean section is completely different, related to these major bacteria. So the conclusion is that yes, it's possible to modify the infant microbiota after birth, but it's very important to notice this result does not indicate that all mothers with cesarean section should provide a fecal sample for their babies. And why so? Because it's also possible that the infants get some harmful bacteria associating with later disease development when transplanted. And this is an area that definitely needs more research. This slide summarizes a study on mothers with inflammatory bowel disease and their babies. Mothers had dysbiosis related to their disease, and their infants also presented dysbiosis when compared to healthy infants. Controls had more bifidobacteria and less proteobacteria that tend to increase in dysbiosis. In this study, the researchers also showed that the baby poo acted as an immunostimulatory agent in a mouse model and promoted inflammatory reactivity in mice. However, it's too early to say whether the dysbiosis detected in these infants was permanent or whether these children are at risk for chronic diseases or not. Another question is whether dysbiosis in the gut is linked to any clinical symptoms in an infant. The answer is that it's possible that there are some symptoms. This is a study showing that infants that cried a lot, almost 3 hours per day, had less diversity in their stool samples. But as you see, there is considerable variation and overlap when compared to controls. And at individual level, it's difficult to categorize the infants based on the diversity of their gut microbiome. Dysbiosis in the gut may affect the health of an infant also in another way. And it's been stated that dysbiosis as such may prolong the period when a child is susceptible for getting a disease. This example is based on bronchiolitis, that is the most severe respiratory infection of babies and caused by respiratory syncytial virus and major cause for hospitalization of babies. And unfortunately now, after this corona pandemic, we are experiencing a real epidemic of bronchiolitis in many countries. This infection affects mostly babies up to 3 to 4 months of age, but if a baby has dysbiosis, the vulnerable period may extend up to 8 months. In the next slide, there is another example on how microbiota may have a link to disease development in children. This study investigated children with high genetic risk for type 1 diabetes. Intriguingly, the researchers observed a change in the microbiota of the children more than a year before the clinical diagnosis of the type 1 diabetes. But one does not have a definite answer whether the immune system had altered and this change in gut microbiota was resulting from that, or whether this microbiota change induced the immune system in that way that the disease became clinically detectable. When thinking about factors that may affect infant microbiota, antibiotics are the ones with most drastic effects. Here you see how amoxicillin, marked with yellow line, has a long-lasting effect on microbiota, up to several weeks or even months. Amoxicillin is the most common antibiotic used in young children. In some children, the effects on microbiota resemble those seen in chronic inflammatory conditions, although it's not visualized here. But here on the left-hand side, you see how results on microbiota research are reporting. So it's needs a lot of expertise also to interpret the results in different publications. So microbiota may be perturbed in many ways, but do we know how to correct dysbiosis and what actions or changes would be beneficial? There are unfortunately no definite answers. The available probiotics that have been on the market have a minor effect, and some of them may even be harmful. And it's difficult to define normality. Here you see on the right-hand side bacterial counts of different bifidobacteria and how these counts change along the first years of life. But there is no answer to the question which of these bifidobacteria would be the most beneficial ones. Are some of them harmful? And should we use some of them as supplements if a baby has dysbiosis? And could this dysbiosis be corrected by giving these bifidobacteria? I did not touch nutrition and diet here and the impact of nutrition in shaping the microbiota. The purpose of this particular slide is to show you the complexity of nutritional assessment. And please do not go into the details. It's just not the diet, but also the metabolites that play a role. And in breastfeeding, also the genetics of the mother that have an influence on how the gut microbes in an infant work and how they are fed. So this was a brief overview on infant microbiota and possible links to child health. What next and what to wait for? I guess we need improved technology for more rapid and efficient analysis and means to conduct clinical studies. There are also interesting studies going on related to therapeutic responses and microbiota and whether we could predict therapeutic responses by analyzing the microbiota and detecting non-responsive and responsive factors in the microbiota. But what we are definitely needing are functional studies to be able to learn how microbes interact with our immune system. At our university, we have an ongoing prospective study on the development of microbiota, and this study we have managed to recruit about 1,000 infants that have been followed up now for 4 to 5 years. And hopefully we will learn more about the normal variation and be able to detect some predictive signs for disease in the next few years. I thank you for your interest, and I'm looking really forward to the following discussion.
There are two very interesting presentations, and unfortunately Professor Kolho won't be able to join the discussion because she had a technical problem with her computer, but I think Professor Viemann will do the discussion with me, and I have here some questions. And maybe, do you want to begin with the audience or some of those that have been submitted online? Is the same? So we have here very interesting questions because this is really a thrilling topic. Mostly these are for you. Okay? Do these discoveries mean that S100 proteins can have a role both as biomarkers of sepsis, but also as a potential therapeutic immunomodulatory tools? So it would be my question as well. Should we use them in clinical practice in a way to trigger a more powerful and more perfect response to inflammation? This is what is a potential therapeutic role.
Yes. Of course, I mean, it's my personal opinion from this point of view, but definitely, even at my former university in Hanover, it took me a while to convince the attendings. But in Hanover, we use it already.
In the neonatology
in
units?
the neonatology department, we determine, and it halved the use of antibiotics.
And as we see,
this is amazing.
No,
and
amazing.
These are hard certain attendings, and they are now. I thought they never leave their concept, but they do it, and this is successfully. And this is why we just were granted in Germany a preclinical study to use it as diagnostic, and also then for therapeutic intervention. Yeah, because it's a double situation. We can use it to define the intervention groups better by measuring it, filtering those out that might profit from it, not to treat it like every preterm infant's now needed. No, that's not the point. Yeah, but we think this is possible, and in Hanover it was very successful so far.
This is really amazing, because if we listen to what Professor Kolho said, when you use antibiotics, and sometimes we must use because there is neonatal sepsis, we are inducing dysbiosis. So
exactly.
If you are using less because you promote, you trigger immune response, you could use less antibiotics.
Yeah,
I think
absolutely.
It's important. So the next question is, considering these discoveries, could you use artificially synthesized calpoprotectin, of course, as 100 proteins as a therapeutic tool to help mature neonatal immune system? In a way, you just answered it.
Yes.
Now, moving to the adult, could these proteins have a role in regulating inflammatory gut diseases and inflammatory associated tumors of the gut, like colorectal cancer? Could they be used therapeutically to prevent and control these conditions?
Okay.
You
even said obesity?
Adults. No, it's not here.
Not obesity.
No.
Because it's all inflammatory bowel disease, obesity, and so on. And in this study, we even showed that this priming with S100s, these cohort, we followed up until the third, fifth year of life. And if you have a deficient situation in the neonatal period of intestinal S100, they put on weight exceptionally, so they get these obese children. So this is very amazing. But to come back to this question, the role of calprotectin
in the adult to prevent colorectal cancer,
adults,
for example, or other bowel diseases.
As you know, especially in adult IBD, calprotectin is used as a perfect marker, the monitoring marker for diagnosis and also for the treatment concept, then whether you are successful or not. I think, and now it comes a clue there, we try to explain it in a review. S100, if you have high levels in a non-inflammatory situation, then it's not a damage-associated molecular pattern, then it induces tolerance by pre-priming, by pre-activating the. I think S100 is one of the biggest NF-κB suckers. So they just induce a pre-activation, then NF-κB shifts into the nucleus and is not available for next stimuli. Okay, if it's released due to cell death, like this is a definition for all DAMs, also called alarmins, then it's an amplifier in this situation. When it comes together, then they all hook on this MyD88 dependent pathway, and then you have inflammation like crazy. So this, it's a question of time, of intervention. Yeah, if we would know that this is a risk person for IBD in the future, I don't know whether in the pre-stage one could change something. But anyway, my personal opinion is that the establishment of the microbiome takes place, the fixed microbiome, during the first year of life. Everything afterwards causes whatever we, and especially my colleague from Helsinki talked about, are transient shifts. If you stop it, it reverses to exactly the fixed microbiome you had at 1 year of life. So this is amazing. So if you drink buttermilk for a month, yeah, you will change. It will switch us back again.
Good. This is good news. Now, considering your both works, would it be possible that some of the benefits of fecal transplantation will be due to the effects of S100 proteins present in feces like calprotectin? So, it's mixing your two talks.
Whether it would be a good idea to transplant fecal samples with high S100 levels, yeah, might be a synergistic effect. I guess, yeah, that would be clear. Yeah.
Now I have a question for you. Very recently, I was reading about the fact of female immune system to be more reactive, more powerful than the male immune system. Namely, for example, if we take the reaction to a COVID vaccine, it seems that women were reacting more powerfully than men. So my question is, and it is true that autoimmune diseases like multiple sclerosis, rheumatoid arthritis are much more common in women than in men, about 80%. So my question is, are there any noticeable differences in immunity between male and female when they are born? So the level of S100 alarmins would be different in the female or the male sex, so that females are reacting more powerfully, more effectively than male babies. Because there was something in your slides with 0.77, a paper from 2021, that male seems to have more alarmins. So this comes in my doubt. What is are they different when they are born?
Yeah.
And why are they different when they are born, according to sex?
According to sex, okay. This could also be a question for Professor Ghana. So it's a polygenic, poly-environmental thing at the end. Like, so everything. This is the easiest answer I can give here now. Yeah, because of many aspects. As neonatologists, we know if we are called into the delivery room, like a preterm birth, 24th week, the first question we are posing when we are running, male or female?
We do the same as obstetricians.
Yes,
it's a female. Well, she will do better.
We relax already.
Yes, she will do better.
Running, yeah?
It's true.
So males really have a disadvantage, preterm males at least. And we have actually, I don't know where you saw some of this data, but we didn't present it. They have at birth lower levels, and this explains why they are so risky for fatal causes of sepsis, in my opinion. But hey, it's, of course, I sell also this story. I think there are other host factors connected to this, involved in this perinatal period that are important as transient regulatory mechanisms. Okay, it's not only about S100. So we were lucky just to see this, that this might be an overall concept, and if such regulatory mechanisms are lacking, you are prone to overshooting inflammatory responses, which at the end causes fatal outcomes. Yeah? And their males have a drawback. And in my opinion, we confirm knowledge that has been known for centuries by midwives and whatever. Now we just reveal the molecular mechanism. Because vaginal birth, labor, this is what induces this release of S100s in the mother and also in the child. Yeah? And so this is a very good idea. Whether it's so I doubt that it's only, and this is probably where we are not conform, Professor Kolho and myself, it's not only the transmission of vaginal bugs in this vaginal delivery situation, it's also endogenous host factors that act in the same manner on innate immune receptors and that are only released when labor is going on. This is it's not funny, but it's necessary.
I understand, and I agree. But of course, as you said, this is more vast. What I mean by this, it's not only the mode of delivery, but it's the mode of feeding and its diet later on. And well, there are so many factors, and I think, well, at least as an obstetrician, that most cesarean sections have a reason to be done, yes, because there is protracted labor, because there is fetal distress, and so on. So there are more. And breast milk, for me, it will be something very important, very advantageous for the child.
Absolutely, I think, and there was a Cell paper very recently out that. Feeding is extremely important, and breastfeeding. So very new data that supports the situation, this opinion. So there was a nice study done in 2017 when they tried to, in mixed models, with all these factors listed, who has really impact. And actually, for example, mode of delivery, it always depends what time period you look at. Yes, mode of delivery, big impact in these microbiota states in the first 4 weeks of life, say it. If you look afterwards, that doesn't have so imprinting.
Yes, because you are breastfeeding at least for 6 months. So it's a more sustainable effect.
Exactly, is a very imprinting effect on what fixes your microbiota establishment. And a little bit like with this sexual behavior, you just hit a little bit this topic. There are situations when you have to do a CS. It is like this. There are situations when women are not able to breastfeed. Yeah? And so if you do these discussions here and you bring out such data like vaginal delivery is important, breastfeeding is important, you make women like this and families feel bad.
Feel guilty.
Grandfathers, grandmothers start to, hey, my daughter-in-law, she doesn't want to do breastfeeding and so on. So big discussion. So we also have to be carefully how to communicate such data. This is important, yeah.
And not to be fundamentalists.
Yeah, exactly. That's why. And yes, for example, in such situations, S100 substitution also is something that we can offer them. This is also the idea behind.
I don't know if there is any question from the audience. They are very convinced with your talk. This is good. So I think we could close the session, and I'd like to thank you again for coming and giving us such a nice lecture and giving us new path for the future.
Yeah.